In our timeline, we will show you Mendel's generation to our generation. From Mendel's generation in 1822 to 1930, grandparents' generation in 1930 to 1960, parents' generation in 1961 to 2001, and our generation from 2001 to 2017.
From 1822 to 1930, there were several very important discoveries regarding genetics, so I'm going to tell you a few that I thought were very interesting. Gregor Mendel was a famous scientist who lived from 1822 to 1884. He was most well known for his work with pea plants, and he cross and self-pollinated these plants to discover many important concepts such as genotypes, phenotypes, and dominant and recessive traits. He completed these studies at the University of Vienna. In 1838, Matthias Schleiden discovered that all plants are made of cells. He did so by studying the structures of plants under a microscope. He was educated at Heidelberg University, and he also recognized the importance of the nucleus, which was first discovered in 1831 by Scottish botanist Robert Brown. Last but not least, Walter Fleming described mitosis in 1879. He began his studies at the University of Göttingen and continued them at Tübingen, Berlin, and Rostock. He was mentored by Franz Eilhard Schlues, who himself was strongly influenced by Max Schlues, who was one of the first cell biologists ever. Friedrich Meister first isolated DNA in 1969 under the study of Felix Hoppe Seiler.
In 1943, during World War II, Max Delbruck and Salvador Luria began the study of bacteriogenetics by employing bacteriophages, a type of virus that invades bacteria, to learn about the bacterial genome. This information led to advances in learning about diseases that can cause harm to the human body. In 1953, Francis Crick and James Watson studied the double helix structure of DNA. Their experiments led to conclude that the structure of DNA meets the requirements for a substance that encodes the genetic information for an organism. In 1960, Francis Crick, Francis Jacob, and Jacques Monod began study of what was known in molecular biology as the missing messenger. The missing messenger was to learn how DNA translated information to proteins. Through their studies and research, they conducted experiments and learned that the missing messenger was a form of RNA called messenger RNA. Messenger RNA received information from the DNA and carried it to proteins.
In 1961, Marshall Nirenberg, a young biochemist at the National Institute of Arthritic and Metabolic Diseases, discovered the first triplet, a sequence of 3 bases of DNA that codes for one of the 20 amino acids that serves as the building blocks of proteins. By 1966, Nirenberg was able to crack the entire genetic code and found all 64 possible triplets. In 5 years, Marshall Nirenberg cracked the entire genetic code. Molecular biologists by the 1970s had discovered the genetic code and could spell out the sequence of amino acids in proteins. But there was an inability to read off the precise nucleotide sequence in DNA. In 1977, Walter Gilbert and Frederick Sanger worked separately in the United States and England, developing new techniques for rapid DNA sequencing. Both scientists found separate methods to sequence DNA, and the combination of these methods led not only to the sequencing of DNA, but them winning the Albert Lasker Medical Research Award in 1979, and how they got the Nobel Prize in Chemistry in 1980. A microscopic worm, commonly used in genetic studies, was the first multicellular organism to have its genome sequenced. The worm, called Caernarhabitus elengus, lived in soil and grows to be a millimeter in length. The worm's body is translucent, so researchers can closely observe biological development as the worm grows from a fertilized egg to an adult with exactly 959 cells. Three scientists were awarded the 2000 Nobel Prize in Physiology or Medicine for discoveries made independently using C. elegans. Sidney Brenner, John E. Solston, and H. Robert Horses were honored for their work on how genes control the development of organs and the death of cells in the worm and since it transferred over into human bodies.
In 2001, first draft of the human genome sequences were released simultaneously by the Human Genome Project and Cellular Genomics. In 2004, Merck introduced a vaccine for human papillomavirus, which promised to protect women against infection with HPV 16 and 18, which inactivates tumor suppressor genes and together cause 70% of cervical cancers. In 2007, Michael Worobey traced the evolutionary origins of HIV by analyzing its genetic mutations, which revealed that HIV infections had occurred in the United States as early as the 1960s. In 2008, Houston-based Introgen developed a Devex, utilizing a form of adenovirus to carry a replacement gene coding for the p53 protein. In 2016, a genome was sequenced in outer space for the first time, with NASA astronaut Kate Rubins using a Menayon device aboard the International Space Station.